Today I want to talk about design, but not design as we usually think about it. I want to talk about what is happening now in our scientific biotechnological culture, where for really the first time in history, we have the power to design bodies, to design animal bodies, to design human bodies. In the history of our planet, there have been three great waves of evolution. The first wave of evolution is what we think of as Darwinian evolution. So, as you all know, species lived in particular ecological niches, in particular environments, and the pressures of those environments selected which changes through random mutations in species were going to be preserved. Then human beings stepped out of the Darwinian flow of evolutionary history and created the second great wave of evolution, which was we changed the environment in which we evolved. We altered our ecological niche by creating civilization. And that has been the second great couple 100,000 years, 150,000 year flow of our evolution. By changing our environment, we put new pressures on our bodies to evolve, whether it was through settling down in agricultural communities, all the way through modern medicine. We have changed our own evolution. Now we're entering a third great wave of evolutionary history, which has been called many things: intentional evolution, evolution by design, very different than intelligent design, whereby we are actually now intentionally designing and altering the physiological forms that inhabit our planet. So I want to take you through a kind of whirlwind tour of that, and then at the end talk a little bit about what some of the implications are for us and for our species as well as our cultures because of this change. Now, we actually have been doing it for a long time. We started selectively breeding animals many thousands of years ago. And if you think of dogs, for example, dogs are now intentionally designed creatures. There isn't a dog on this earth that's a natural creature. Dogs are the result of selectively breeding traits that we like. But we had to do it the hard way in the old days by choosing offspring that looked a particular way and then breeding them. We don't have to do it that way anymore. This is a beefalo. A beefalo is a buffalo cattle hybrid. And they are now making them, and someday, perhaps pretty soon, you will have beefalo patties in your local supermarket. This is a geep, a goat-sheep hybrid. The scientists that made this cute little creature ended up slaughtering it and eating it afterwards. I think they said it tastes like chicken. This is a kama. A kama is a camel-llama hybrid, created to try to get the hardiness of a camel with some of the personality traits of a llama, and they are now using these in certain cultures. Then there's the liger. This is the largest cat in the world, the lion-tiger hybrid. It's bigger than a tiger. And in the case of the liger, there actually have been one or two that have been seen in the wild. But these were created by scientists using both selective breeding and genetic technology. And then finally, everybody's favorite, the zorse. None of this is photoshopped. These are real creatures. And so one of the things we have been doing is using genetic enhancement or genetic manipulation of sort of normal selective breeding pushed a little bit through genetics. And if that were all this was about, then it would be an interesting thing. But something much more powerful is happening now. These are normal mammalian cells genetically engineered with a bioluminescent gene taken out of deep-sea jellyfish. We all know that some deep-sea creatures glow. Well, they've now taken that gene, that bioluminescent gene, and put it into mammal cells. These are normal cells, and what you see here is these cells glowing in the dark under certain wavelengths of light. Once they could do that with cells, they could do it with organisms. So they did it with mouse pups, kittens. And by the way, the reason the kittens here are orange and these are green is because that's a bioluminescent gene from coral, while this is from jellyfish. They did it with pigs. They did it with puppies. And in fact, they did it with monkeys. And if you can do it with monkeys, though the great leap in trying to genetically manipulate is actually between monkeys and apes, if they can do it in monkeys, they can probably figure out how to do it in apes, which means they can do it in human beings. In other words, it is theoretically possible that before too long, we will be biotechnologically capable of creating human beings that glow in the dark. Be easier to find us at night. And in fact, right now, in many states, you can go out and you can buy bioluminescent pets. These are zebrafish. They're normally black and silver. These are zebrafish that have been genetically engineered to be yellow, green, red, and they are actually available now in certain states. Other states have banned them. Nobody knows what to do with these kinds of creatures. There is no area of the government, not the EPA or the FDA, that controls genetically engineered pets. And so some states have decided to allow them, some states have decided to ban them. Some of you may have read about the FDA's consideration right now of genetically engineered salmon. The salmon on top is a genetically engineered Chinook salmon using a gene from the salmon and from one other fish that we eat to make it grow much faster using a lot less feed. And right now the FDA is trying to make a final decision on whether pretty soon you could be eating this fish. It'll be sold in the stores. And before you get too worried about it, here in the United States, the majority of food you buy in the supermarket already has genetically modified components to it. So even as we worry about it, we have allowed it to go on in this country, much different in Europe, without any regulation and even without any identification on the package. These are all the first cloned animals of their type. So in the lower right here, you have Dolly, the first cloned sheep, now happily stuffed in a museum in Edinburgh. Ralph the rat, the first cloned rat. Cece the cat for cloned cat. Snuppy, the first cloned dog. Snuppy for Seoul National University puppy, created in South Korea by the very same man that some of you may remember had to end up resigning in disgrace because he claimed he had cloned a human embryo, which he had not. He actually was the first person to clone a dog, which is a very difficult thing to do because dog genomes are very plastic. This is Promedia, the first cloned horse. It's a Haflinger horse cloned in Italy, a real gold ring of cloning because there are many horses that win important races who are geldings. In other words, the equipment to put them out to stud has been removed. But if you can clone that horse, you can have both the advantage of having a gelding run in the race and his identical genetic duplicate can then be put out to stud. These were the first cloned calves, the first cloned gray wolves, and then finally the first cloned piglets, Alexa, Chrissy, Carol, Janie, and .com. In addition, we've started to use cloning technology to try to save endangered species. This is the use of animals now to create drugs and other things in their bodies that we want to create. So with antithrombin in that goat has been genetically modified so that the molecules of its milk actually include the molecule of antithrombin that GTC Genetics wants to create. And then in addition, transgenic pigs, knockout pigs from the National Institute of Animal Science in South Korea are pigs that they are going to use, in fact, to try to create all kinds of drugs and other industrial types of chemicals that they want the blood and the milk of these animals to produce for them, instead of producing them in an industrial way. These are two creatures that were created in order to save endangered species. The guar is an endangered Southeast Asian ungulate. It was a somatic cell, a body cell was taken from its body, gestated in the ovum of a cow, and then that cow gave birth to a guar. Same thing happened with the mouflon, where it's an endangered species of sheep. It was gestated in a regular sheep body, which actually raises an interesting biological problem. We have two kinds of DNA in our bodies. We have our nucleic DNA that everybody thinks of as our DNA, but we also have DNA in our mitochondria, which are the energy packets of the cell. That DNA is passed down through our mothers. So really what you end up having here is not a guar and not a mouflon, but a guar with cow mitochondria, and therefore cow mitochondrial DNA, and a mouflon with another species of sheep's mitochondrial DNA. These are really hybrids, not pure animals. And it raises the question of how we're going to define animal species in the age of biotechnology, a question that we're not really sure yet how to solve. This lovely creature is an Asian cockroach, and what they've done here is they've put electrodes in its ganglia and its brain, and then a transmitter on top, and it's on a big computer tracking ball. And now using a joystick, they can send this creature around the lab and control whether it goes left or right, forward or backwards. They've created a kind of insect bot or bug bot. It gets worse than that, or perhaps better than that. This actually is one of DARPA's very important. DARPA is the Defense Research Agency, one of their projects. These Goliath beetles are wired in their wings. They have a computer chip strapped to their backs, and they can fly these creatures around the lab. They can make them go left, right. They can make them take off. They can't actually make them land. They put them about 1 inch above the ground, and then they shut everything off, and they go boom. But it's as close as they can get to a landing. And in fact, this technology has gotten so developed that this creature, this is a moth. This is the moth in its pupa stage, and that's when they put the wires in and they put the computer technology so that when the moth actually emerges as a moth, it is already pre-wired. The wires are already in its body, and they can just hook it up to their technology, and now they've got these bug bots that they can send out for surveillance. They can put little cameras on them, and perhaps someday delivering other kinds of ordnance to war zones. It's not just insects. This is the Ratbot, or the Roborat, by Sanjeev Talwar at SUNY Downstate. Again, it's got technology, it's got electrodes going into its left and right hemispheres, it's got a camera on top of its head. The scientists can make this creature go left, right. They have it running through mazes, controlling where it's going. They've now created an organic robot. The graduate students in Sanjeev Talwar's lab said, is this ethical? We've taken away the autonomy of this animal. We'll get back to that in a minute. There's also been work done with monkeys. This is Miguel Nicolelis of Duke. He took owl monkeys, wired them up so that a computer watched their brains while they moved, especially looking at the movement of their right arm. The computer learned what the monkey brain did to move its arm in various ways. They then hooked it up to a prosthetic arm, which you see here in the picture, put the arm in another room. Pretty soon the computer learned, by reading the monkey's brain waves, to make that arm in the other room do whatever the monkey's arm did. Then he put a video monitor in the monkey's cage that showed the monkey this prosthetic arm, and the monkey got fascinated. The monkey recognized that whatever she did with her arm, this prosthetic arm would do. And eventually, she was moving it, and eventually stopped moving her right arm, and staring at the screen, could move the prosthetic arm in the other room only with her brain waves, which means that monkey became the first primate in history of the world to have 3 independent functional arms. And it's not just technology that we're putting into animals. This is Thomas DeMars, the University of Florida. He took 20,000 and then 60,000 disaggregated rat neurons. So these are just individual neurons from rats. Put them on a chip. They self-aggregated into a network, became a integrated chip, and he used that as the IT piece of a mechanism which ran a flight simulator. So now we have organic computer chips made out of living self-aggregating neurons. Finally, Musa Avaldi of Northwestern took a completely intact, independent lamprey eel brain. This is a brain from a lamprey eel. It is living, fully intact brain in a nutrient medium with these electrodes going off to the sides, attached photosensitive sensors to the brain, put it into a cart. Here's the cart, the brain is sitting there in the middle, and using this brain as the sole processor for this cart, when you turn on a light and shine it at the cart moves towards the light. When you turn it off, it moves away. It's photophilic. So now we have a complete living lamprey eel brain. Is it thinking lamprey eel thoughts sitting there in its nutrient medium? I don't know. But in fact, it is a fully living brain that we have managed to keep alive to do our bidding. So we are now at the stage where we are creating creatures for our own purposes. This is a mouse created by Charles Vacanti of University of Massachusetts. He altered this mouse so that it was genetically engineered to have skin that was less immunoreactive to human skin, put a polymer scaffolding of an ear under it, and created an ear that could then be taken off the mouse and transplanted onto a human being. Genetic engineering coupled with polymer physiotechnology coupled with xenotransplantation. This is where we are in this process. Finally, not that long ago, Craig Ventnor created the first artificial cell, where he took a cell, took a DNA synthesizer, which is a machine, created an artificial genome, put it in the cell and of a different cell. The genome was not of the cell he put it in, and that cell then reproduced as the other cell. In other words, that was the first creature in the history of the world that had a computer as its parent. It did not have an organic parent. And so, asked the economists, the first artificial organism and its consequences. So you may have thought that the creation of life was going to happen in something that looked like that. But in fact, that's not what Frankenstein's lab looks like. This is what Frankenstein's lab looks like. This is a DNA synthesizer, and here at the bottom are just bottles of A, T, C, and G, the 4 chemicals that make up our DNA chain. And so we need to ask ourselves some questions. For the first time in the history of this planet, we are able to directly design organisms. We can manipulate the plasms of life with unprecedented power. And it confers on us a responsibility. Is everything okay? Is it okay to manipulate and create whatever creatures we want? Do we have free reign to design animals? Do we get to go someday to Pets R Us and say, look, I want a dog. I'd like it to have the head of a dachshund, the body of a retriever, maybe some pink fur, and let's make it glow in the dark. Does industry get to create creatures who in their milk and their blood and their saliva and other bodily fluids create the drugs and industrial molecules we want and then warehouse them as organic manufacturing machines? Do we get to create organic robots where we remove the autonomy from these animals and turn them just into our playthings? And then the final step of this, once we perfect these technologies in animals and we start using them in human beings, what are the ethical guidelines that we will use then? It's already happening. It's not science fiction. We are not only already using these things in animals, some of them we're already beginning to use on our own bodies. We are now taking control of our own evolution. We are directly designing the future of the species of this planet. It confers upon us an enormous responsibility that is not just the responsibility of the scientists and the ethicists who are thinking about it and writing about it now. It is the responsibility of everybody because it will determine what kind of planet and what kind of bodies we will have in the future. Thanks.